Listo, pues vamos a comenzar formalmente nuestro seminario del día de hoy. El día de hoy tengo el placer de presentarles al doctor Sanyog Dwivedi, que él, bueno, es miembro de nuestro departamento. Él obtuvo su licenciatura y maestría en biotecnología por la Universidad de Amity en India. Desarrolló o ha desarrollado aquí en México una serie de investigaciones, primeramente en el Departamento de Biomedicina Molecular del CIMBESTAF del Instituto Politécnico Nacional en el área de inmunología del cáncer para obtener el grado de doctor. Actualmente en nuestro departamento es becario postdoctoral de GAPA y él trabaja con la doctora Patricia Rendón Huerta y con el doctor Luis Felipe Montaño. Él es experto y ha sido un estudioso de la asociación inmunitaria tumoral, la supresión inmunitaria de las células NK y el agotamiento inmunológico en el cáncer gástrico. Ha presentado sus resultados en diferentes congresos nacionales e internacionales y también ha publicado sus investigaciones en diversas revistas internacionales. Y bueno, como el doctor Dwivedi es extranjero, aunque tiene ya unos años viviendo aquí en México, él nos va a presentar el día de hoy su seminario en inglés. Va a tratar de hacerlo lento, despacio, para que todos podamos entenderlo. Pero bueno, creo que cualquier duda que tengamos, pues podemos dirigirnos a él para que nos la aclare. Adelante, doctor.
Thank you for having me, and thank you everyone for joining here. Also, I'm grateful for this opportunity to present my work. And so I'll start my presentation with a good afternoon to everybody. And let me share my screen. Okay, so thanks for the introduction, and I am going to present my work in English, but I hope I'll try to speak slowly. But if you have any questions, you can ask me in Spanish. I will try to understand as best I can. If not, I'm around anyways. We can discuss anything. So most of my work revolves around, Claudin6 and natural killer cells. And some of it is more turned toward Claudin6, and rest of it are actually intermingled to each other. So I will try to, put them in sections and go through them one by one and slowly. So start with the cell-to-cell junctional complexes, which are, multi-protein. Sorry. I'm just looking for a marker. Yeah. So I'll start my work with cell-to-cell junctional complexes, which are basically multi-protein cellular structures responsible for adhesion or cell-to-cell contact. There are four more important junctional complexes that we have known so far, starting from the apical most tight junctions, following adherent junctions or desmosomes or gap junctions. Also, some non-classic junctions and focal adhesion hemidesmosomes also has been discussed, but the more important ones are tight junctions. Like I told, they are multi-protein, so tight junctions are basically made up of more than 40 different proteins of different categories, including adapter proteins or signaling proteins or scaffold. Importantly, including also transmembrane proteins like junction adhesion molecules or occludin and isolines. And then comes the claudins, which are transmembrane, and also they form the backbone of tight junctions. Claudins are usually of 20 to 34 kilodalton size, and they are usually present in epithelia and endothelia. The more important thing they provide is they make a paracellular barrier among cells, and also they provide ports for the passage of different solutes through them. So basically the tight junction permeability is a unique feature which was provided by claudins. If we check at their structure, almost all the claudins have transmembrane domains they have these extracellular loops, which are called EL1 and EL2. EL1 is a little larger and is responsible for this paracellular ion selectivity. And EL2 is responsible for oligomerization, and which means like the bonding between Claudin and Claudin, which could be among the same Claudin or with the other Claudin. On the cytoplasmic side, they have a N-terminal and C-terminal, and C-terminal has this carboxy terminal and the PDZ interacting domain. Also, they have sites for phosphorylation and palmitoylation, which is actually necessary for their proper folding and proper structure patterns and localization as well. Also for downstream signaling. So usually the Claudins have, they have this general structure, but different Claudins, and depending upon how close they are to each other, or how similar they are to each other, or depending upon how similar to the foundation structure, they are grouped into not classic ones, where you can see they show structural similarity. Like here you can see Claudin6 and 9, which are very similar. And I work with Claudin6, while other Claudins also share a structural similarity. While there are others which are little less similar than the foundation or among each other, which are grouped into non-classic Claudins. And irrespective of their structure, and when we talk about Claudins expression, it start getting interesting. Different Claudins and their expression, and more importantly, their cellular localization, is tissue specific, which is more relevant in terms when we talk about cancer. So why is that? Because many of the Claudins, or most of the Claudins, actually express usually embryonic or fetal tissues, and then when the cell transform, it's also linked with the aberrant expression of Claudins in the cells, which according to the tissue, which may be upregulation, downregulation, or merely the mislocalization of Claudins, and that contribute to different aspects of cancer progression. So either case, the expression and the regulation of expression of Claudins is regulated by multitude of signaling starting from different ways. And once they are there and they are expressed, or whichever location they are, they further affect, or they control or regulate or induce different pathways or things. Like here we can see they affect apoptosis or barrier function, cell cycle regulation, tumorigenesis, EMT, and so on. So the multi-dollar question is, do Claudins promote or suppress cancer? And trust me, it gets, every time we talk about a different Claudin in different tumor, it becomes very interesting to understand how they are actually a part of cancer progression. So answer to this question lies in three different kind of interactions that Claudin can provide to cells. First is when they are properly located or not located, how they affect the paracellular sealing function. The secondly, which proteins they interact or not interact affect further downstream signaling and trigger certain pathways. And the third is the direct interaction. So when Claudins are properly located and they perform the paracellular sealing function, they prevent the passage of several things. But when they are not in their place, they allow the passage of microorganisms, growth factors, toxins, which further enters through paracellular passage and triggers inflammation, which is like a pre-process of tumor initiation. The second, certain proteins like PDK, you can see, or YAPTAZ or beta-catenin, which are known onco promoters, once they are attached with Claudin. So if Claudins are properly placed, they attach with these proteins, so then don't let them to get activated and trigger certain pathways. But when they are not in place, these proteins are actually free, they get activated, and they trigger certain pathways that actually leads to inflammation, proliferation, survival, EMT status, and several more, just to promote cancer progression. The third one is the interaction of Claudin directly with certain protein or protein complexes. Let's take a look at here. I mean, there are several interactions that Claudin can establish with protein or protein complexes, which triggers multitude of downstream signaling, interconnected or not. Some we know, many of them we don't know yet. We're still exploring. And like here you can see Adam Chan interaction with Claudins, or integrin integration like we saw in the previous slide, MMP interaction also with certain adhesion molecules, or indirect interaction with some cytokine receptors. All this leads to activation of further downstream pathways and activates activity of certain transcription factors and triggers transcription of several genes, which directly and indirectly promote cancer progression. So like I said, so this regulation of Claudins is actually part of disruption of cell-to-cell junctional complexes, which in turn is a part of even greater phenomena called epithelial mesenchymal transition, in which these junctional complexes compromised cell undergo cytoskeletal remodeling, and which allows them to attain this migratory back front polarity. And now these cells are actually free to invade or migrate, and they are way more capable in many senses to survive and to promote cancer. So like we discussed before, it is very interesting more in terms of which Claudin we are talking and which tissue we are discussing, and then we are looking at cancer perspective. So it can either be upregulation or downregulation, or both of in the same tissue of different Claudin that can promote cancer. And this is a small list from my thesis, my doctorate thesis, and you can see several of the Claudins are upregulated or downregulated, or in some tissues like prostate, there's few which are downregulated and promote cancer, and some other which upregulate and promote cancer. And I basically work with gastric cancer and more on Claudin6, which is the topic of today's presentation. So more than a decade ago, in our lab, Dr. Erika herself, she published a paper, and we saw that Claudin6, along with 7 and 9, they actually are highly upregulated in gastric cancer, different histological types of gastric cancer. So when we talk about gastric cancer, 90% of gastric cancer are adenocarcinomas, and till 2020, it has been reported they are fifth in fifth place in number of new cases, or fourth in terms of number of mortalities. So still they are very valuable. And this is one of the very interesting paper came in the beginning of 2011, where these cells which we work, gastric adenocarcinoma cells called AGS cells, when these cells were transfected with Claudins, different Claudins, importantly Claudin6, 7, and 9, what we saw, most of these Claudins, some way that they localize in membrane, but a lot of it actually localizes in cytoplasm, which is not a usual place for Claudins to be. But this mislocalization and overexpression of Claudins led the cells towards increased invisibility, and also gave them a higher proliferation. This is a recent paper that came from the lab, I think last year, where we also saw that these Claudin6 transfected AGS cells have high expression of CD44, which is a cancer stem cell marker. Interestingly, it also we also noticed a CD24 negative, CD44 positive population, which increased to 1.4 folds with transfection of Claudin6 in AGS cells. At the same time, we also noticed several markers of cancer stem cells, and also the EMT, which we found is highly upregulated in these transfected cells. So in basics, what it tells about Claudin6 expression or expression in gastric cancer. So first, we know there is with the increased expression of Claudin6, there is higher transmigration, invasiveness, and proliferation. Also, they are more tend towards stem cell-like properties, which assures cancer progression. But there are a lot of information that we don't know, and to find that information, we also needed a little more tools than just working lab. And that is about the classification. Like, we have no information how many gastric cancers are actually positive for Claudin6, or even they are overexpressing Claudin6. And if that is the case, where lies these Claudin6 overexpressing gastric cancer in conventional and new molecular classifications? The second is how it fits in the chart with the survival and prognosis compared to other well-known or previously known properties of or survival characteristics of gastric cancer. Also, we needed to find out the associated genes or mutations or specific molecular signatures. And more importantly, how is the immune interaction? Is it a specific, something specific we can figure out, or is it just a casual or something similar to some other molecular subtypes or groups? So in 2019, I found this amazing study from a Japanese group, which where they proposed Claudin6 as a single prognostic marker and a tumor-promoting gene in subgroup of intestinal type gastric cancer. And that led us to the quest, and we recently published this paper where we identified some interesting thing about Claudin6 positive gastric cancer. So Claudin6 overexpressing gastric cancer in molecular classification. In general, if we talk about classification of gastric cancer, the most discussed and clinically significant that has always been discussed is histological classification, which could be either intestinal type, which is usually well differentiated, or diffuse type with poor differentiation, or a mix of both. But what I'm more interested in to talk about the molecular characterization of gastric cancer, which was given by the Cancer Genome Atlas, and they proposed 4 different molecular subtypes depending upon the molecular signatures gastric cancer possess. So here you can see the first and actually the most important one, which actually holds a maximum number of gastric cancer cases, is chromosomal unstable type of gastric cancer, which usually has intestinal type histology, and they have higher p53 mutations. Following that, the second and actually more aggressive category, which is called genomically stable, which holds diffuse histology, and they have CDH1 and Rho A mutations, along with fusion between Claudin18 and ARH gap. Following, there is epistine virus positive with PIK3CA mutations and PDL1 overexpression, also CDKN2A silencing. Also, then microsatellite unstable, which has hypermutation, and also we have MLH1 silencing. So to finalize the study, I used the Cancer Genome Atlas program and their data, which I accessed using this web program called CBio Portal, and then I analyze Claudin6 expression. So as you can see, the first thing that actually comes here, and it looks interesting in that sense, that not all the gastric cancer patients or samples have Claudin6 expression. So it's not universal, though only 34% of gastric cancer patients have Claudin6 overexpression, which also includes a small percentage of mutations, including missense and truncating mutations, that the significance is still unknown. However, when I check the molecular signature of these 34% Claudin6 overexpressing gastric cancer samples, I found they possess molecular signatures of chromosomal instability. So 96.88% basically belong to this molecular subgroup. However, there is a small percentage which still have a molecular signature or a mixed of genomic stable molecular subtype. In comparison to Claudin6 low cancer, which actually distributed into several groups in evenly like 41 to 13, 27 in different groups. The next question, we compared the survival. We did a survival analysis, and we found that the overexpression of Claudin6 in gastric cancer actually tends to poor overall and also progression-free survival in comparison to Claudin low, and also another analysis we also compared it with different groups. So actually Claudin6 is a more severe subset of CIN, which we started thinking from here. So as you can see, Claudin6 overexpression, and this we already know, and cancer progression, and we have now little information about classification and survival prognosis. So what about the associated genes or specific molecular signature? We did a mutation analysis of Claudin6 highlight low groups, and then we checked, and we found some interesting results. First, as a signature mutation of CIN group, Claudin6 high gastric tumors also have higher mutations in p53. And the following three genes, MIEN1, STAR-D3, and PGAP3, are actually part of ERBB2, which is a very interesting thing to look on, which I'm working right now. But they also have higher mutation, and mostly it's amplification in similar group of patients. And following, there is higher mutation CCNE1. However, the other two proteins, the ARID1A and DOG3, are more prominently mutated in Claudin6 low group. Besides higher mutations, it's also interesting to see the MEN1, STAR-D3, and PGPA3 are actually also overexpressed in Claudin6 high group samples. Interestingly, even though p53 has higher mutations, it is slightly downregulated in Claudin6 high gastric cancer. However, there is no significant difference between DOG3 and ARID1A. Like I said, these three, MIEN, STAR-D3, and PGPA3, are part of ERBB2 amplification group. So I wanted to check what is the expression of ERBB2. And interestingly, there is a high correlation between Claudin6 high gastric cancer and ERBB2 expression, which you can compare to other groups, which I have included in almost all the future slides you're going to see. So Claudin6 high and low, you already know. This is total samples with chromosomal instability in the TCGA database. This is total samples of the database. And this one is a little interesting. This is CIN group sample, but without Claudin6 high group sample. So some places you're going to see a significant difference when we remove Claudin6 high group. And there is a difference that is contributed to the chromosomal instability group, especially because Claudin6 high is a subset of that. And then we identified differentially expressed genes associated with Claudin6 overexpression. We find 1,316 upregulated genes and somewhat around 100 downregulated genes. These upregulated genes are specific to Claudin6 high overexpression gastric cancer, which you can see when we compare to other groups. These genes are specifically overexpressed in this with Claudin6 overexpression. Importantly, also with the overexpression of these genes actually tends to poor overall survival of patients in gastric cancer. Then to find some specific markers which are like more attached to Claudin6 overexpression, I have seen that several apolipoproteins and MAGIA genes, melanoma antigen gene family A, and several of the members of this family are, I cannot say exclusively, but they are exclusively upregulated in Claudin6 high gastric cancer, and also is equally true for apolipoproteins. And I wanted to know, with the upregulation of these specific genes, which pathways or which biological processes are highly affected, because they have a great significance of understanding the prognosis of gastric cancer, especially with the Claudin6 high gastric cancer. So when we did a gene set enrichment analysis of these genes, we found that there is high upregulation of cholesterol metabolism and protein digestion absorption. Also interesting that these Claudin6 high gastric cancer are more capable of drug metabolism, so the drug will be cleared fastly than the other subsets. Interestingly, there is a collective downregulation of several pathways involved in innate and adaptive immunity. Interestingly, Th1 and Th2 cell differentiation, also B cell receptor signaling pathway and toll-like receptor signaling pathway. So what this tells us, in conclusion, in nutshell, we can see that Claudin6 overexpressing gastric cancer, besides what we already know, have intestinal histology and CIN molecular subtype. So it is considered now as a subset of chromosomally stable molecular subtype with intestinal histology. And the enrichment analysis also confirmed that these Claudin6 overexpressing gastric cancers have rewired metabolism, effective drug clearance. Also, they have poor overall and proportionality survival. And more importantly, they tend to have suppressed innate and adaptive immune responses. And to my interest, I wanted to look more in this section from now onward, especially the markers related with NK cell cytotoxicity. So some basics about how NK cell or cytotoxic activity against cancer works. So there are two major cytotoxic cells which actually infiltrate tumor cells, and then they can kill tumor. However, how they recognize tumors is different, but how they kill tumor cells is pretty similar. So I'm going to forward with the NK cell. So they basically work with NK cells have a bunch of markers on their surface, which can be categorized into activating receptors or inhibitory receptors, depending upon which function they serve. So if the engagement of receptors activate NK cells, so they are activating receptors, or if engagement of receptors, refrain the cell to get activated, they are inhibitory. And usually the NK cell activation depends upon a collective signaling, how much of engagement of activating versus inhibitory happen, and that what decides the fate of NK cell, if they're gonna get activated or not. So what happens when NK cell come in contact with a healthy cell? The engagement of inhibitory receptors with MSC class 1 molecules triggers NK cell not to get activated because it's a healthy cell. But on the other side, when an NK cell engage with a tumor cell or an infected cell, the engagement of activating receptors activates NK cell. But when this happens, let's take a closer look what exactly happens with the activation of NK cells. So first thing is the NK cell forms a conjugate with target cell, and this process is called formation of immunological synapse. When this happens, it's followed by engagement of several activating receptors. If I have to name someone like 2B4 and KG2D, which is one of the most important activating receptors, and another group which is called NCRs, natural cytotoxic receptors, NKP30, 44, and 46, and some others. So engagement of activating receptors leads to a downstream signaling, which finally produce IP3, which is a calcium messenger, and triggers calcium flux. With the calcium flux, triggers MTOC polarization, which basically means the MTOC will allow the movement of lytic granules towards the immunological synapse. And this process is called lytic granule polarity towards immunological synapse. What happens next? With the engagement, now NK cells are active and they can kill a tumor cell in plenty of ways. The first one is more important that we discussed, that this granule polarity will release cytolytic granules here in the synapse, which have granzyme B and perforin. As you can see here, perforin makes pore in the membrane and allows the enzyme to enter in the cancer cell, which further degrade everything it finds, and then cell dies. More than that, NK cell can also kill a cancer cell by engagement of death ligand receptors, which you can see here, Fas ligand and TREA, which are expressed in NK cells. But when they engage with their receptors, they trigger apoptosis using caspases. The other one, and which is also very important, is the ADCC, antibody-dependent cell cytotoxicity. And what happens here is the CD16 is capable of binding with Fc receptor antibodies. Now, these antibodies are already present or bound to the antigens on cancer cells, and NK cell identifies that and kill further the cancer cell. Also, it's supplemented by cytokine secretion like interferon gamma and TNF alpha. Now, this is a brief summary of how a NK cell can kill a cancer cell. And if they are capable of killing cancer cells, then why still cancers exist? And that's $1 million question. So it's a simple picture of some important activating receptors we saw in the previous slide, some inhibitory receptors, and also some co-activating receptors. And what happens with the progression of cancer, they develop tumor microenvironment, which are the most interesting thing in today's cancer research. We know is the accumulation of different type of cells and the cytokines, messengers, or other molecules that actually are secreted by this calcium niche, which triggers suppression of immune cells, especially cytotoxic cells, and in terms now we are talking about NK cells. So here in the bigger picture, it's easier to understand that tumor cell take measures for NK cell to be incapable of not killing a tumor cell. And here you can see first thing they do shed several important ligands that can activate or that can engage and activate NK cell activation receptors. The second, they also engage several immunosuppressive cells, which secrete a different number of messenger molecules or chemicals to make NK cell, to suppress NK cell to get activated. And third, they also upregulate inhibitory receptor ligands that engage with inhibitory receptors and let not NK cell to get activated. So this collective phenomena where NK cells are in the tumor and they are not able to kill cancer cells is called the existence of exhausted NK cells in the tumor microenvironment. So to understand if in Claudin6 high gastric cancer has also do something with these markers of NK cells, which are responsible for activation. I further did analysis with this TCGA pan-cancer atlas data, and here I make a group with overexpression of Claudin6 depending upon their mRNA scores, more than 5 or less than 1, and that led to Claudin6 high or low group creation. So here, you can clearly see the Claudin6 high gastric cancers have down regulation of NKG2D expression in comparison to low gastric cancer tumors. And similarly, NKG2 ligand, ULBP1, are quite significantly upregulated, which is a common feature of several cancers. I also checked several natural cytotoxicity receptors, and the important one, NKP44, which can be seen highly downregulated in Claudin6 high gastric cancer. Also, NKP46 is highly downregulated in comparison to Claudin6 low gastric cancer. In cytolytic granules contents, when we did a comparison of Claudin6 high and low gastric cancer tumor, there's several of them, including Fas ligand, trail, and also perforin, which is found to be downregulated. However, LAMP1 overexpression, which is interesting and also being noticed is get upregulated in several different cancers, and which we will come to in later slide also. The other co-stimulatory molecules of NK cells and their ligands also found to be downregulated, like you can see here, C244 and DNAM1. If you remember, we have seen them in the previous slide of general receptors where I presented to you. And here, Nectin-2 and PVR2, both are actually upregulated in general, which is a common characteristics of cancer. Then in the last group, I wanted to introduce you to CD16, which we have seen is sponsored for ADCC through NK cells. However, there is a associated molecules, which is also necessary for NK cell cytotoxicity, is significantly downregulated in Claudin6 high gastric cancer. Though the CD38 receptor PECAM1, which is a marker of endothelial cells, doesn't show any significant change here. So in a nutshell, we can conclude it like this, like what we have seen so far, that there is increased Claudin6 expression in AGS cell and transfected them, which is associated with several aspects that we have discussed before, the transmigration, invasiveness, or proliferation. On the second side, we have been seeing in previous slides that there is reduction of NK cell cytotoxicity or exhausted NK cell population, and which leads to existence of creation of exhausted NK cell population. But how this all looks in lab, if we want to see how it is going in our model. So one of the previous results we had in this stimulation model, where we use this Claudin6 or Claudin9 transfected AGS cells, and then we stimulate NK cells, which in this case was purified peripheral blood NK cells. And when we stimulate these NK cells for 4 hours with these transfected cells, and then we analyze several markers on the membrane, what we have noticed that there is significant downregulation of Fas ligand and LAMP1 on Claudin6 and 9 transfected NK cells, which were stimulated with these AGS cells. So what this means? What is LAMP1 significant in this case? So this is, if you remember this image we just discussed. So when NK cell get activated, this downstream signaling and then calcium flux. With this calcium flux, what happens? The cytoskeleton rearrangement triggers polarity of cytolytic granules towards immune synapse. Well, these cytolytic granules have LAMP1 and 1, 2, and 3 on their membrane, and more importantly, LAMP1. They also have Fas ligand on them. Also, they contain granzyme and porphyrin. And when this granule goes and fuses in the membrane and releases the content in the immune synapse, the LAMP1 on the granules actually stays on the membrane. And that is why LAMP1, CD107A or LAMP1 is considered a degranulation marker of both NK and T cell. So the question is why there is reduction in LAMP1 in our model which we are working. And behind this conclusion, there is a whole lot of theory, but I will try to give something that actually start making sense to you. So this is amazing research, I think came in 2015 by a Chinese group, where they proposed this phenomena, which includes C38 that I introduced you in previous slide. And this C38 in NK cells has a direct correlation with degranulation of NK cells. They basically propose a C38 is essential for degranulation, and when that happens, we can see LAMP1 on the membrane. So what is CD38? So CD38 is a multifunctional protein, which is also an antigen, at the same time is also an active enzyme. What it does with this enzymatic activity depends on it's happening in which pH, acidic or basic. But at the end, it produces some specific molecules called ADPR or cyclic ADPR and ATP. And all 3 of them are calcium messengers and responsible for calcium mobilization. So this group, they clarified this phenomenon, which includes NK cell activation by engagement or stimulation with tumor cell. In general, there are other phenomena, but when NK cell get activated by the engagement of tumor cell, it triggers a downstream signaling and activates CD38 to be localized on cytolytic granule. And when once it is there, it produces ADPR, which we saw here through the enzymatic activity. This ADPR triggers TRPM2 calcium channels and allows calcium mobility which is responsible for cytolytic granular polarity towards synapse. So it's pretty clear here that CD38 is essential for granular polarity following tumor cell stimulation, and then for degranulation, and that's how we're going to see LAMP1 on the membrane. And this is the result from the same paper, but what we see here is in presence of CD38, when NK cell forms a synapse with a tumor cell, the perforin and enzyme are perfectly delivered to immune synapse. However, in absence of that, the same phenomena doesn't happen, and the perforin and enzyme are dispersed somewhere in the NK cell. And without NK cell, without CD38, there is no degranulation. So in presence of CD38, you see a good expression of LAMP1 on the membrane. However, this doesn't happen without CD38. So it's pretty clear here that there is no CD38, then there will be no ADPR generated within the cytolytic granule. So the TRPM2 won't get activated, no calcium mobilization, will stop the tumor-stimulated degranulation, and there will be no LAMP1 on the surface. So I wanted to test this phenomena in our model, and I wanted to see how this is happening with us, and what is the picture of in terms of this granular polarity and CD38, and in general NK cell immune suppression in our model. So there are two important questions. First is when we put NK cells and we stimulate them with the tumor cell, and in our case it is Claudin6 or with LPS stimulation of wild type AGS cells, that leads to two important questions. Either there is a direct receptor ligand signaling, or some secreted component from AGS cells interact with NK cells that may or not lead to reduction in CD38 activity, and that is why we have a reduction in LAMP1 on NK cell surface that we saw in a previous result from our lab. So firstly, I wanted to take out the possibility if these NK cells are getting stimulated because of some secreted component from AGS or Claudin6 transfected cells. So what I did, I take the AGS or Claudin6 transfected cells, and then I took the conditioned media and I stimulated the NK cells from 4 to 24 hours, different experiments, and then I measured CD38 expression through cytometry. And I haven't seen any significant difference among different NK cells stimulated with either GFP or wild type or with Claudin6 transfected AGS cells. Then I also wanted to be confirmed that the CD38 activity, if by chance we are seeing, is not because of its ligand interaction, which usually, if you remember, I mentioned about PECAM1, which is also called CD31, and usually it expresses on endothelial cells, but not on epithelial cells. However, it has been noticed in some gastric cancer, some cancers, that the epithelial cell also express CD31. So I wanted to be sure that CD31 is not expressed in our model, and as you can see, there is no expression in general in AGS cells. Then this is still a work in progress, so I just wanted to show some things here. And we were having some trouble with the Claudin6 transfected cells lately, so I wanted to test my hypothesis in another alternative approach, and where it's a previous paper from the lab where we have seen the LPS from H. pylori after 24-hour stimulation also triggers expression of several claudins, including Claudin6, 7, and 9. So the idea is still the same. I wanted to check how the granular polarity or NK cell suppression is working out. Post-stimulation is just instead of Claudin6 transfected cells, now I'm using these wild type cells with LPS stimulation. So these AGS wild type cells, first I stimulate them with LPS 10 nanogram per ml for 24 hours, Then these AGS cells with wild type versus wild type plus LPS stimulated, I use them to stimulate NK cells for 4 hours, and then I take out the NK cells and I do expression analysis to see what is the expression of CD38 or other markers of NK cell cytotoxicity. So here what you are seeing that, please let me clarify so don't get confused. So even though these marked as wild type or wild type LPS, I'm actually basically talking about NK cells which are stimulated with either wild type or wild type LPS. So all the results down below are also the same NK cells. They are stimulated with these cells. So what we see here that the CD38 positive NK cells without stimulation, almost all the cells are CD38 positive, and there is slight downregulation of CD38 expression in the cells, or the frequency of CD38 positive cells is slightly less once it is stimulated with wild type or wild type plus LPS. But there is not significant difference between them. And these results are also, some of them are contributed by my intern in the lab, Ana Paula, which is helping with the repetitions of some experiments. So if you see here, just remember. So first thing we saw, there's slightly less positive cells. However, when we check the expression of CD38 post-stimulation with wild type or wild type plus LPS treated cells, we saw there is a significant downregulation in the membrane expression of CD38. However, if we have a difference between them, yet is not concluded, but in many cases we have seen the effect, whatever effect we are seeing after cancer stimulation in NK cells is just a little more severe with LPS stimulation. So we are still figuring out, but you can see usually there is a little more severe than the only wild type cells. So in general, there is downregulation of CD38 membrane expression. Later on, we checked LAMP1 expression on NK cells. So first thing that happens, the stimulation with AGS cells increases the LAMP positive cells, which makes sense because when AGS cell or cancer cell come in contact with NK cell, they should start granulating. So there is granulation, that's why there is LAMP1 in both the cases. You can see the 34.9 to 34.6 in comparison to 26.9% LAMP positive cells. However, though there are more LAMP positive cells, the overall LAMP expression on the membrane of these cells is way less than NK only. So there is significant downregulation of LAMP1 expression on membrane of NK cells. So to conclude, you can see that there is downregulation of CD38 on the membrane, and also there is less LAMP1 on the membrane, even though there are more positive cells for LAMP1. Following, I wanted to check the intracellular CD38 expression following stimulation with these cells, AGS wild type or AGS wild type stimulated with LPS for 24 hours. And sorry for this hodgepodge image, but it was interesting and important to see how this high yellow expression is actually changing between populations. So these are controls, and here NK only CD38. What do you see if you count the overall CD38 positive cells? They don't change significantly from 98 to 96 or 94.9. But what is interesting, if we divide the population in terms of high or low CD38, then in NK only, there's not too many cells which are actually with CD38 low expression, and most of them are actually in the high side. However, when NK cells are stimulated with wild type or wild type plus LPS stimulated AGS cells, there is clear demarcation of high and low expressing populations. And what tells it, like this from 74.5, after in contact with the cancer cells, so many cells, somewhat around 20% cells, actually downregulated to low CD38 low population. And the same thing happens with the LPS stimulation as well. Whereas there is a population which was high before, and then it turned to low expression of CD38. And here I compared the intracellular CD38 expression with the stimulation the same way with wild type or wild type LPS. And then on NK cells, we see there is a clear downregulation of CD38 intracellular expression from NK only to wild type or wild type LPS stimulation. And as I told you before, usually the impact is a little severe here, but is not significant yet. We are doing with the repetitions, maybe with the more number of expression, we experiment, we can confirm that. Secondly, we check the LAMP1, and it's a similar experiment. And when NK cells are stimulated with NK only control or with the wild type or wild type LPS ATS cells, there is clear demarcation of the clear existence of two different populations, the high and low. And when we do the partition, you can see something really interesting here. In the NK only, actually most of the cells have low LAMP1 expression, and very few are actually high, like 11.7% only. But when they come in or contact with the cancer cells, the expression of LAMP1 actually increases, and there is a huge population. I mean, from 11.7 to 14.9% now expressing a high levels of LAMP1 intracellularly. And here also you can see with the LPS stimulation, the ratios are the same, but clear existence of two populations due to cancer stimulation. This is the LAMP1 expression in terms of geometric mean fluorescence intensity, which also clearly states the overexpression of LAMP1 intracellularly in NK cells after stimulation with wild type cells. But like I said, we are still is a work in progress. So we're still looking for a statistical significance. So the picture till here, what it tells you that these are NK only, and there is LAMP1, there is CD38, there is LAMP1 or CD38 on the membrane, and this structure is supposed to depict cytotoxic granule, but we are not sure about the localization yet. So let's just keep it like this. So what we see here, in the previous slide, we see there is downregulation of CD38 on membrane, less LAMP1 on membrane. Now we see less CD38 intracellularly, but more LAMP1 intracellularly. And this is the results I have to conclude, but it's also very clear that I checked the perforin, which is a cytotoxic granule content, and when we checked the perforin positive cells intracellularly, we saw 93.8% cells, NK only, are perforin positive. However, once these cells were stimulated with wild type or wild type LPS ATS cells, we saw there is a clear downregulation or the clear decrease in positive populations. And when I check the mean fluorescence intensity, there is a significant decrease in NK cells treated with wild type or wild type plus LPS. Like I said before, usually this is more severe, but however, difference between them is not significant, but is a clear decrease in perforin concentration. So to conclude this lab work, this last section, what we have seen so far that the NK only have perforin enzyme, CD38, LAMP1, and also cytotoxic granules. And following the previous thing that we have discussed, also there is decrease in perforin intracellularly. This is the last concluding image of this presentation. So I'm just going to tell my hypothesis and what we are working in, what we need to look forward in in following days. First is there is clear downregulation or decrease in C38 on membrane, which is followed by the decreased expression generally, or decreased presence in cytoplasm. So there is less C38 here, and that's why there is less C38 on the membrane, which seems clear, but C38 existence and its presence on membrane or in cytolytic granule is actually very complicated thing to understand. But linearly, if you see if there's less in the cytoplasm, there's less on the membrane. So it seems a straight line to me. However, we have seen there is less lampon on the membrane, but there is more lampon in the cytoplasm. So there could be several possibilities. First, the cancer cells are actually activating NK cells, but due to reduced cytolytic granule polarity that we have assumed, it doesn't reach to membrane even though after activation. However, it's another question to see and to understand if the increased lampon in the cytoplasm doesn't load in the granules first place, or it loads sufficiently in the granules and doesn't go to the membrane. So that we still need to see. And that's a similar question for perforin. However, it is reduced here. And so we have probably gonna see a less secretion of it when we check the cytotoxicity by cytosat assay. However, we don't know if it is how much of it is going to load in the granules and how much will be transported to synapse. So like I said, it's a work in progress, and we have to look for these questions. These days, I'm on to this localization of. I'm checking the localization of these molecules intracellularly, and soon we have answer of these questions if they don't load or they load and they don't go to the membrane significantly. So with this, I will conclude my presentation. Thank you so much, and I want to say my thanks to the GAPA and also Faculty of Medicine and obviously NAM, which they have given me a lot of opportunities here. And I also want to thank my mentors, Dr. Arika Rendon and Dr. Luz Felipe Montaño, and my amazing team members, which are quite few, both Maris, which actually provides so many things and provide system that we work in. They maintain their Chris, Juliana, which was an intern there, and Charlie, who's about to finish his PhD from the lab. Verania was also another intern, and Jorge, and Naresh, and obviously me. So thank you so much.
Muchas gracias, doctor.
Your microphone is off.
Oh, listo.
Okay.
Muchas gracias, doctor, por esta excelente ponencia y, bueno, por esta muestra del trabajo que están realizando también pues en el laboratorio en donde usted está realizando estas interesantes investigaciones. Quisiera dar pues pocos minutos para si alguien tiene alguna pregunta o algunas palabras para el doctor. Después con Pedro.
No se oye.
Su micrófono, doctor San Pedro. Ahí está.
Ah, ya, es que no escuché bien.
No escuché.
Perdón. Bueno, pues muchas gracias por la exposición de un tema tan interesante. Para mí, pues soy lento, un tema muy extenso, mucha información, pero en forma resumida, ahora sí que voy a hablar en español lento. Mi inglés de negro de Harlem probablemente no sea muy adecuado, pero pues ni modo. El problema fundamental, hasta donde entiendo, es que de la familia tan amplia, 24 a 27 tipos distintos de vacunas, bueno, se ha estudiado la sobreexpresión de Claudin6, que tiene muchas consecuencias, pero una de ellas, que parece ser el punto central de esto, es el bloqueo en la actividad de las células NK, entre otras muchas cosas. Y esto me parece muy interesante porque estaba por preguntar por qué estamos o están estudiando las alteraciones moleculares consecuencias de la sobreexpresión de Claudina 6 una vez que ya se estableció. Y esto me parece muy importante porque, a reserva de que desde luego no, el cáncer de estómago no existe, sino que existe una gran variedad de cánceres de estómago. En el caso particular de los epiteliales, las variantes histológicas, como nos presentaste, de tipo intestinal, pues son las más comunes. Y ahí resulta que el consenso general, hasta donde tengo entendido, es que la asociación es clara con procesos inflamatorios previos, o sea, una gastritis común, una difusa, una folicular o incluso una gastritis atrófica. El detalle es que la sobreexpresión de Claudina 6 también se ha encontrado e incluso parece ser más intensa en los cuadros inflamatorios, más intensa que en el 34-36% del total de casos de cáncer gástrico, como nos presentaste. Y si nos vamos al tipo intestinal, bueno, pues entonces resulta de gran importancia el asunto previo, es lo que estaba por preguntarte, pero tú mismo lo contestaste. Con este trabajo de Montaño y Rendón, de Luis Felipe y de Erika, por ahí la vi, donde hacen un estudio sobre el papel del proceso lesivo de Helicobacter pylori sobre TLR2, y eso genera la sobreexpresión, se relaciona con la sobreexpresión de la Claudina 6. En ese sentido, pues me parece que eso debería tal vez comentarse un poquito más porque le da toda la secuencia, es decir, hay elevación de sobreexpresión de Claudina 6 en condiciones de proceso inflamatorio y el más importante, pues sin duda, es el Helicobacter pylori, como tú mismo lo pusiste. Eso está padrísimo, pero a lo mejor me perdí porque estaba atendiendo también a otra charla, perdón, pero me parece que eso le daría más cuerpo, porque es algo que tu propio grupo ya ha trabajado. Y la otra cosa, solo dos dudas. Acá está. Nos hablaste de la localización citoplásmica de Claudina 6, que normalmente pues, se encontraría asociada a la membrana. Entonces, cuando nos ven unas muestras ahí con inmunofluorescencia, que pues, obviamente no tienen la resolución de estudios ultraestructurales, pero no sé si hay algo detallado, porque uno supondría que dada la estructura de la Claudina 6, uno esperaría que si está dentro del citoplasma, debe estar en algún sistema de membrana. Entonces eso sugeriría que esa internalización se debió al sistema endosómico. Entonces ahí mi duda es si se sabe algo al respecto. Y la última es si la sobreexpresión de Claudina 6 se asocia con esto, con cáncer, básicamente de estómago, aunque también de otros tipos de cáncer. Bueno, uno supondría que es un blanco interesante en inmunoterapia, pero por alguna razón parece ser que los investigadores se han ido más por la Claudina 18.2. ¿Tú sabes algo al respecto? ¿Por qué dejaron la Claudina 6 y ahora están más concentrados en Claudina 18.2? Por lo demás, muchas gracias y felicidades por me da mucho gusto verte y te felicito por este importante trabajo, que en resumen es, pues nada más agrégale lo de la inflamación que pues, Montaño y Rendón mostraron, y entonces ya al menos yo le vería más congruencia. Y dos, si se sabe algo de la localización específicamente en membranas intracitoplásmica de la Claudina 6, y tres, ¿por qué de repente cambiaron a la Claudina 18.2? Gracias, ya me callo.
Thank you for the question. Sí, puedo responder en inglés, por favor, porque es muy difícil para mí.
Of course, my friend, you can.
So as I understood, there are three questions. The first you ask is why, if it is already established that there is NK cell suppression in general in cancer and gastric cancer, then why I am studying the same? So as per my understanding, there is no established link so far with the Claudin6 overexpression and the immune suppression in gastric cancer. I mean, in general, in either other cancers either. So far, if I understood properly, or if I know properly, there is no proper establishment between Claudin6 and immune suppression at all. And more than that, there is no information about the cytolytic granule trafficking in Claudin6 or with the H. pylori-based LPS stimulation. When we do that either, we don't know if there is, exists a cytolytic granule trafficking which is get affected. And the third, like I told you, it's a work in progression. I'm still working on this experiment. So in the last slide, if you remember, I'm more interested to see where this suppression is happening. The first things either the LAMP1 or CD38 or perforin is not loading in the granules, or if they are loading and there is not granule trafficking. So that is something not being studied yet, as per my information. And the second question that you asked was, I forget. Well,
about the intracellular localization of Claudin6, I think it will be very interesting to study this with electron microscopy, or some other media, so we can find even the chemical structure, the three-dimensional structure of Claudin6. One would assume it will be in some kind of membrane. So this suggests that the internalization of Claudin6 expression is regulated via endosomic system or something like that.
This is like when you were crafting your question, you mentioned that there are 23, 24, or 27 so far known Claudin6, depending on which organism we are talking about. But in terms of cancer, if you're just talking about humans, these 23 different Claudins, they express in terms of tissue, a specific manner, and in different tissues has. And I totally understand. It's very easy to understand if there is down regulation of Claudins, it promotes cancer progression, because now there is less tight junctions, or they're not proper tight junctions, and that's why it promotes cancer. It makes total sense. But the other part, and which is so far as we know about the Claudins in general, not just Claudin6, the upregulation is equally important as their mislocalization. So not just Claudin6, there are other Claudins which also has been seen in different places, including cytoplasm, also Claudin1 supposed in nucleus, and they have been localized in different places in the cell, and they have different impact. One probable hypothesis, because there is no, as per my understanding, there is no direct link so far, but like as a hypothesis, suppose several proteins I showed you, like PDK or beta-catenin, they are on membrane, they are part of tissue, tight junction, but once they are free in the cytoplasm, they are, oncopromoting, they promote cancer progression, or they travel to nucleus, they bind with other proteins, and they do different effects. So how Claudin6 fits in the picture, and there are several studies which have reported, as from my understanding, but how Claudin6 does directly or indirectly that, or how it contributes in the cancer progression in the cytoplasm or in attachment with, other proteins or protein complexes, is open for, research and discussion. And we are a small group, we are doing our best to give those answers. And lastly, like you asked about Claudin18, which is, it's basically characterized in or more associated with the genomically stable gastric cancer or diffuse type gastric cancer. So I don't know if it is right to say, or I'm sorry I'm commenting in this way, but I think people are still studying Claudin6. Our group is studying, and almost every year there are 5-6 new papers that we find from Claudin6 in gastric cancer or in general. I mean, there are some papers I published you, most of them are from last year. I mean, from 2022. So I won't say that Claudin6 research is not happening. I think it's happening, but like you see, it's from a small group. Like we have proposed that it's a subset of that. So I don't think people are working. I think it's working. But gastric cancer, diffuse type gastric cancer, is more aggressive of gastric cancer. So I think that definitely gets more attention in some senses. So that's what I have to say. Thank you.
Thank you very much. One last thing, what attracted my attention is I think it's a fact that the expression of Claudin6 is, we say, normal in embryonic tissue, but in mature tissue, it is down-regulated. So if in gastric mucosa, subjected to inflammation and later to cancer development, is overexpressed, it's some kind of carcinoembryonic antigen or some kind of, so it could be a marker of prognosis. Don't you think?
That has been proposed already, one of the papers I showed you, and also we are onto this because we are, I mean, in the paper we published last year, I identified a bunch of other genes which actually, are highly correlated with Claudin6 expression in gastric cancer, which we are testing in the lab and with our model also these days. So definitely it's a prognostic marker, and definitely it can be a drug target also. I mean, there are some studies, if I remember correctly, they are checking a possibility to target Claudin or some Claudins or Claudin6 also in gastric cancer. And more importantly, like you said, embryonic to adults and then aberrant expression in gastric cancer. But it is more related with, because, this Claudin6 high gastric cancer has stem cell-like properties, and also they have high EMT. And Claudin6 directly and indirectly linked with these processes. So that makes total sense that even though it's aberrantly expressing, but somewhere it is putting cancer or making cancer cells more stem cell-like, and that make them more aggressive.
Muchas gracias.
Thanks for your question. And actually, in general, the whole idea of my work, I mean, I work with 2 different cells. And then in gastric cancer, then it's a specific type of gastric cancer. And then I want to study how that Claudin6 high gastric cancer, or in general, overexpressing gastric cancer has the immune, effect on immune system. So the whole idea of my study is is quite broad, because Claudins are very broad in general. Gastric cancer is very heterogeneous. And then I'm working with NK cells, which is another complexity. So I understand that the presentation was like really broad in sense of explanation, and also it's a work in progress. So not everything, like I was not able to put everything to explain everything. So thank you for your question.
Thank you very much. Congratulations.
Thank you. Thank you.
Bueno, pues muchas gracias a todos los participantes en este seminario. Gracias, doctor Dwivedi, por su presentación. Y bueno, yo quisiera de último compartirle y compartir con todos este reconocimiento al doctor por esta excelente ponencia, y le mandan algunas felicitaciones a través del chat. Muchas gracias, estuvo muy interesante y realmente muy completo, aunque todavía falta por aclarar bastante del panorama, pero muchas gracias por habernos compartido y pues gracias a todos, gracias al resto de los asistentes y bueno, nos vemos en el siguiente seminario en el mes de abril. Muchas gracias y que tengan feliz descanso de Semana Santa.
Thank you, everyone. Thank you
Hasta
for
luego,
joining.
muchas gracias,
Nos
ponente.
vemos, nos
Gracias.
vemos. Adiós a todos, pórtense bien.
Hasta luego.